Hello, everyone. We see some participants starting to log on. All lines are muted and we will begin here shortly. Thank you all for joining us. All lines are muted upon entry and we will go ahead and get started here shortly. Hello, everyone, and welcome to the Foundation Fighting Blindness National Chapter Vision webinar. My name is Bailey Bostelman, and I'm the Regional Field Director for the East here at the Foundation. We're so excited that you will be joining us today, and thank you for sharing your Saturday with us. I'd like to briefly review some logistical details for the call. Currently, all participant lines are muted. Today's call is being recorded and will be shared with all registrants after the meeting concludes. You will also find the recording on the Foundation Fighting Blindness website within the coming days. If you are using a screen reader, please be aware the controls are at the bottom of the Zoom interface. This control bar may collapse when not in use, so make sure to joggle your mouse before clicking anything. If you prefer to prevent the controls from auto hiding, go to your settings within the Zoom platform, select Accessibility, and then select Always Show Meeting Controls. If you have questions throughout the presentation, please send them using the Q&A function within Zoom. Today, our expert speakers will share the most recent research findings, discuss exciting clinical trial opportunities, and highlight the significant impact these trials have on the fight against blinding diseases. At the end of the presentations, we will open the floor for Q&A from our panelists. Whether you're a patient, caregiver, or healthcare professional, we hope this webinar offers valuable insights. Now I'm pleased to hand it over to our first speaker of today's webinar, Amy Laster, who is our interim Chief Scientific Officer at the Foundation Fighting Blindness. Amy, the floor is yours.
Thank you, Bailey. Good day. Again, I'm Amy Laster, and it is my pleasure to be with you today. I have a few slides today, but I will describe the relevant content. Just pull those up. Okay, here we go. Great. All right, I'm just gonna go back to my screen. Give me 1 second. Bear with me here. Okay, great. So you might be familiar with me oftentimes sharing with you information and highlights about the Foundation's science grants and award program. And the goal of our funding strategy really is to drive research toward preventions, treatments, and cures for retinal diseases. Today, my colleagues and I are going to highlight the research roadmap from discovery and development to clinical assessment, all with the goal of getting therapies on the market and available to patients. To set the stage and context, I'm going to define some terms that we hear and use often. Research. Research can be, and in the context of this webinar's topic, is inclusive of work in research laboratories to create tools, test those tools in cells and animals, as well as collecting clinical data from patients in a clinical setting to diagnose, characterize, or even track the progression of disease. You'll hear later that even clinical trials are research, specifically to test the safety and effectiveness of a treatment in humans. So when you hear lab-based translational research, early preclinical research, clinical studies, and again, even clinical trials, we're still referring to research. For the next few minutes, I'm going to step through what is called the development process for therapy development. Then I'll discuss how foundation funding fits into this process. And finally, I'll wrap up with some recent news in the clinical trial landscape for inherited retinal diseases and dry AMD. Man, Here's my slide. Trying to get my slide to move forward. Sorry. Okay. Did my slide move forward?
And Amy, this is Bailey. We're actually seeing the PowerPoint view versus the presentation view for
Okay,
some
thank you.
reason. I know we tested this, so there we go. Perfect.
Okay. Are
Now
you
it's showing large on the screen. Perfect. Thank
okay?
you.
Are you okay? Sorry about that.
No worries.
I just want to go back. All right. Give me trying to get to the webinar. Get to my notes. Over here. Sorry. I'm going to stop sharing for a moment and talk through these next slides. Okay. So again, what I want to share with you is really what we call the drug development process. And in the development of drugs as well as medical devices, they follow really well established paths to make sure that they're safe and effective when they reach the public. So there are three steps in this process. Dr. Durham, he'll describe the clinical trial step. I'll talk about first, there is a drug discovery process. And this typically is when researchers discover new drugs through new insights into the disease process. And this allows researchers to design a product to stop or reverse the effects of the disease. Now, many test of molecular compounds to find possible beneficial effects against any of the large number of diseases, existing treatments that might have unanticipated effects, as well as new technologies, such as those that provide new ways to target therapies and to specific sites within the retina in order to even manipulate genetic material, as you'll hear sometimes talk about gene therapy or gene editing. So at this stage in the process, generally, if it's a drug, there's like thousands of compounds that might be a potential candidate for development as a medical treatment. And after this early testing, there's usually just a few, if it's a compound or if it's a vector, that looks really promising, and that kind of calls for further study. Now, this phase of research is translational research, and so you'll hear translational research often. Now, once researchers identify a promising compound for development, they conduct experiments to gather information on things like how the therapy, if it's say a drug, it's absorbed or how it's distributed or broken down by the body. Really understanding what are the benefits and how is it actually doing the thing that it's supposed to do? What's the best dosage? How should you administer the drug? So you may hear things like subretinal injections or an oral like a pill or something that you take, or intravitreal. So the best way to get the drug to its target. And then also looking at side effects or adverse events that could be toxic. So we want to understand what those are, as well as how it affects different groups of people. and in terms of what stage of disease that they're in, so if they're younger or if they're older, as well as considerations for how it may interact if there are other confounding factors with the disease. Second stage in this development process is preclinical research. And this is really to assess safety and biological activity. So before testing a therapy in people, researchers got to find out whether it has the potential to cause, again, toxicity or any serious harm. And so this is done first by usually looking in cells or looking in animals. And then the FDA really requires that there is, really good laboratory practices. So this is GLP, and it's just regulation for these preclinical laboratory studies. They set the basic minimum requirement for a variety of research activities, as well as just the protocol for quality assurance that oversight is in place. And usually these preclinical studies are not typically large, but again, this is giving information on perhaps how much a therapy should be, the dose of it, as well as the safety, and if there'll be toxicity. And so after preclinical testing, researchers review their findings and they decide whether the drug should be tested in people. Now, this is where I'll go into the third step in this process, and this is toward an investigational new drug or IND application. So, what is an IND application? Now, while preclinical research answers those basic questions about a drug's safety, it's not a substitute for studies of ways the drugs will interact with the human body. And so, clinical research, which refers to studies or trials that are done in people, as developers begin to design the clinical study, they have to really think about what they want to accomplish with each of the different clinical research phases. And Dr. Durham will talk a bit about this. And so they begin what we call an investigational new drug process that they have to go through before clinical research begins. And then they develop either whether it's a researcher or a sponsor, they'll submit a IND application to the FDA or whatever country regulatory agency before any clinical research can begin. Now, these three steps in the development process, discovery, preclinical, and IND enabling, are really critical and necessary to ensure that a drug or therapy is safe and efficacious. So before I turn the floor over to Dr. Durham, I want to share some specific foundation-funded research projects that have advanced through each of the steps I've just described and are currently active clinical trials. The first is a cell-based therapy developed by Dr. David Gam, who's a world-renowned retinal cell therapy at the University of Wisconsin-Madison. Dr. Gam have received significant funding from the foundation over many years for the development of retinal and photoreceptor cell therapies derived from what is called induced pluripotent stem cells. He eventually co-founded a company, Opsis Therapeutic, to further develop the technologies. For the clinical trial advancement of these technologies, the company partnered with Blue Rock Therapeutics, which is a cell therapy company, and they just recently received authorization from the U.S. regulatory agency, the FDA, to launch a phase 1/2 clinical trial for their therapy called OPCT-001. This is a photoreceptor cell therapy for people with inherited retinal diseases. And the study, it's going to look at several dose levels for the OPCT-001, and it will enroll patients in the United States. Now, another advancement of foundation early funding enabled through the foundation's translational research program is an award to an investigator at the University of California, Berkeley, Dr. Richard Kramer. He has research on what's called photo switches, and these are just small molecules that are designed to restore some vision to people with advanced retinal diseases. Um, and to further develop this technology, the University of California licensed the technology to Kiora Pharmaceuticals, which is a company developing treatments for orphan retinal diseases. Now, Kiora has recently received authorizations from the Australian regulatory agents to launch a phase 2 clinical trial for its therapeutic product. And this phase 2 trial, it'll enroll about 36 patients that have ultra low vision or no light perception from advanced retinitis pigmentosa. And we expect this trial to begin at various sites in Australia beginning in next calendar year. And finally, I will highlight our funding partnership with the USH3 initiative. This is a nonprofit organization that has been advancing research for treatments for USH3, and the foundation funded lab studies to prepare for the trial, again through our translational research. For Usher 3, we partnered together to also have a natural history study, which further supports the work of the initiative, because they've just launched a phase 1 clinical trial in Australia to evaluate both safety and tolerability of a oral drug that's for the treatment of individuals with Usher syndrome type 3. And this trial is the first for USH3, and it will also look at how the drug behaves in the body. And that trial is expected to be completed in 2025. These are only a few examples of clinical advancements ongoing, and I will note that the Foundation's funding early in the therapy development process that I've talked about has really been instrumental in advancing more than 80% of active clinical trials for leading retinal disease indications. So I invite you to subscribe to the Foundation to stay updated on all clinical advancements and visit our website often for current research news, where you can learn more about these and other clinical advancements. So with that, I'll turn the call over to Dr. Durham.
Thank you, Amy. And I'm going to pick up the story right there with clinical and clinical development. I'm going to share my slides. And just a brief outline for my prepared comments for today. I'd like to share with you what are the regulatory standards for efficacy or effectiveness of new treatments. How do we get to a marketed product so it can be available to prescribers and their patients? And if someone wants to learn more about the clinical trial pipeline, where do they go? What are some tools that we have available for us? I think it's interesting and important to realize, at least here in the United States, that the evidence standards for showing that a drug is effective, that is, that it has a favorable benefit risk profile, these have been in our federal law since 1962 with the Food, Drug, and Cosmetic Act. And I'll just read here, and I'm showing some slides, but I'm going to explain the relevant content to you. In the 1962 statute, some key words I'm just going to read out loud. To get a new drug approved, quote, it requires, quote, evidence consisting of adequate and well-controlled investigations, including clinical investigations, on the basis of which it could fairly and responsibly be concluded that the drug will have the effect it purports in the labeling or proposed labeling thereof. So, end quote. So the implications of this, we have to study the new treatments in people. And there needs to be a clinical investigation. It would not suffice to approve a new product for treatment just on the basis of evidence from an animal model, for example. Also, we have to have high quality, that is adequate and well-controlled investigations. That means we couldn't approve a new therapy on the basis of a single study. We need to have multiple studies to demonstrate an effect. And lastly, key implication of this section of the code, is that what the drug is supposed to do should be accurately reflected in the product label and the package insert so the prescriber can make a good recommendation for his or her patients. Obviously, federal law, including this one, is quite broad and it's open to interpretation. Because of this, the United States Food and Drug Administration, or the FDA, has issued several guidance documents over the years to clarify how much evidence is required to get new products approved. In 1998, they defined a term substantial evidence as being at least two well-designed studies that demonstrated a statistically and clinically relevant effect of the treatment, or it could be one very large, well-designed study that demonstrated very persuasive statistical evidence and clinically relevant effects. So if you remember from your statistics class, if a large study was done and had a very small p-value, that would be considered something very statistically persuasive, and it would need to be a clinically relevant effect that would impress an MD enough to suggest prescribing it to a patient. And then in 2019, they further opened up some additional avenues to require this substantial evidence requirement. That could be one adequate and well-controlled study, plus a little bit more in the form of confirmatory evidence. And that confirmatory evidence could be another well-controlled study in another related disease. It could use some real-world data. It could be carefully collected natural history data or some compelling non-clinical data, perhaps in a species that was more closely related to humans. So we have now multiple avenues to get new products approved, but of course, it takes a long time to do it.
Todd,
And
this is Bailey. Sorry to interrupt you. It seems that we are not seeing your slides. So I just wanted to stop you just in case.
Okay.
That way that I think we're having a little bit of technology issues today when it comes to Zoom accepting these slides.
Okay, do you see it now?
Perfect, we do. We are seeing it in the PowerPoint mode versus presentation mode. So whichever you prefer.
Yeah, let me try now. How about now?
Beautiful. Yes.
Okay.
Thank you.
Thank you, Bailey, for stopping me. So the obviously it takes a while to get a new product approved. Now I'm really going crazy on the buttons. And Amy showed you earlier this a slide or explained this pathway from discovery and development to preclinical research. And now I'm going to talk a little bit about clinical research, which answers some key questions like what are the short-term benefits and the risks associated with a new product? And why do I say short-term here? It's because within the context of a clinical trial development plan, we may be only able to follow people for a few years versus when that new product is made available for medical use, we can monitor people for years, if not decades. So that would be a long-term evaluation. And then secondly, we want to know are the benefits of the new product, are they similar across treatment centers and patient subsets? Are there examples where maybe we can find that we would not prescribe a new medicine? And then this package of clinical data goes to the FDA and in the US case, but the EMA and the European case, and there are other regulatory bodies around the world, and they're going to look at all the data that's pulled together from the clinical experience. Now I'd like to just get into a few more details about the clinical development pathway and the phase 1, phase 2, phase 3 differences. And you're probably already very familiar with this. And I've pulled out some information from published data on the typical sizes of phase 1 studies, 2 and 3. Phase 1 studies are typically in healthy volunteers and designed just to look at the initial safety signal and look at perhaps dose ranging, what happens with regard to safety as we increase the dose. In the inherited retinal disease space, we often find that phase 1 and phase 2 are combined because they're rare disease, and oftentimes what we learn in healthy volunteers with inherited retinal disease is not going to be terribly informative as to what we might assume happens in a patient in phase 2. And then the confirmatory phase is in phase 3. And what we do in phase 3 is confirm efficacy signals that we have observed in earlier phases of development. In general, and this doesn't always apply, and I'm going to show you some exceptions, In general, phase 1 studies are quite small. In general, they can range from 20 to 100 participants, whereas phase 2 can range from 100 to 300, and phase 3 in some disease areas can be in the hundreds to thousands of participants. It will come as no surprise to this audience that we're not going to have inherited retinal disease trials of thousands of people. We know that there aren't that many people to support such a plan. The one thing I wanted to highlight, there have been research over the years to try to estimate what's the probability of the new treatment entering various phases will ultimately be approved. And this is kind of difficult research to do because there is no publicly available data set of all the new therapies that have ever been studied to know exactly whether they ever got approved or not. So it's kind of a difficult assignment. But at least one paper that gave some thought to the quality of underlying data estimates that of those products that enter phase 2, only 1/4 of them ultimately get approved. So this is a big point where maybe the safety is not there, maybe it's disappointing proof of concept or early efficacy signal. Whereas in phase 3, according to this paper, about 2/3 of new products that enter phase 3 are ultimately approved. I don't know, this isn't a rare disease research paper, that's just sort of a general estimate. The numbers for rare disease like inherited retinal disease might be quite different. I want to go over just to illustrate a couple of the differences between drug development for rare conditions like an inherited retinal disease and a more prevalent condition like dry AMD or geographic atrophy. I have a slide here. It's just really a copy and paste of the benefit risk considerations when the FDA approved Luxturna for RPE65 related retinal dystrophy. They wrote a summary of really what the clinical evidence was and the case for making this gene therapy available. And as you will all be very familiar with, Luxturna was approved on the basis of the MLMT, the multi-luminance mobility test, which was really demonstrate a person's ability to navigate a course in a room with varying levels of light. And Luxturna showed very, very clearly that with dimmer and dimmer lights, people's treated eyes with Luxturna were able to navigate that course much more easily than in the condition with their untreated eye. It's really the one point I wanted to make here is Luxturna was approved on the basis of a single phase 3 trial of only 31 participants. Whereas recently approved complement inhibitor called pegcetacoplan injection for geographic atrophy secondary to dry AMD, its phase 3 program was 2 studies, each with 620 plus or more participants in each one. So this is a much larger phase 3 program. And this product was approved on the basis of demonstrating that the Syfovre-treated participants had a much slower growth in their lesions than the untreated patients were. And that was the basis of approval. So all of these considerations will have been discussed with the FDA and other regulators before the clinical development plan got too far advanced. So this really, I think, demonstrates how different products can be during development, but also the need for frequent interactions with the regulators to know what's required to be successful at the end of the day. Now let me shift gears for a moment. Many people are curious to know how many trials are there out there for me to consider. I looked at the best starting point I would suggest is the Foundation Fighting Blindness website. Our science team updates this website quarterly, and what I had looked at today was updated and the last time in October. So we'll do it again at the beginning of 2025. So please consult our website. We can provide the link here to you. And as I counted this morning, there are currently 49 clinical trials in the pipeline covering inherited retinal disease and dry AMD or geographic atrophy. But inevitably, if you found a trial on our website and you want to learn more, you will probably have to do a bit more research, and we're obviously happy to be resources for you here at the Foundation. So I would recommend as next steps, either an internet search to look up the company or the sponsor of the trial, number one, to get the latest news. But secondly, clinicaltrials.gov, I would suggest is probably the single best source of the most comprehensive information to find out more about a trial, more than we'll readily have on our hands. And I'm going to give you just an example of how you might use it. And I also want to just acknowledge that clinicaltrials.gov may not be very accessible to individuals who have experienced some vision loss. But I'm going to share with you some of the tools that are available on the clinicaltrials.gov website. When you go to clinicaltrials.gov, there is a search tool that you can enter in. And I entered in last night Stargardt in the condition or disease. And I also clicked a radio button for study status that indicates I'm interested only in studies that are recruiting and not yet recruiting. And then further down in the search engine, I could select study type of interventional. So I want to know about Stargardt clinical trials that are recruiting or soon will be recruiting. That was my strategy here. And when I input those parameters into the search tool, I came up with 7 studies in the results. And the results pages give you a hyperlink that will then allow you to click on each hyperlink and learn more about each individual clinical trial that's listed there. And even from this first page of results, I can see what the study title is. I can see what the number is. The status is it is currently recruiting in Stargardt disease. A second study here is a phase 2 tolerability study and effects of ALK-001. And because I know what ALK-001 is, that's the Alkeus clinical trial that's been featured in our news recently. So just as an example, I click on that link itself, and I can then browse the content that's provided to me about the Alkeus clinical trial. And that subsequent link is where I can find out where the trial is being conducted. And for example, there are 8 locations, investigative sites for this particular trial. And I just on the first couple of results there, I see that Los Angeles, University of California, Los Angeles is still recruiting. And so is Gainesville, Florida, still recruiting for this particular study. And this result would also allow me to find out who could I call with a study sponsor or the sites to learn more if I wanted to participate in the clinical trial. And lastly, for advanced users, I would say of clinicaltrials.gov, this is an FYI. It's possible once you have a search set up in clinicaltrials.gov, you can export the results just like you would any other search tool. And I did that last night and came up with a list of, I wanted to see how many retinitis pigmentosa trials could I find. And I created a downloadable table that would let me see what are the interventions or the studies, the treatments being studied, who was the sponsor, what conditions were being studied. And there are several other fields that I could use, say, as an interested person, perhaps in learning more about it or tracking the progress over time. And I provide this information today just for informational purposes. Just because we list or mention a trial at the foundation, we don't not necessarily endorsing it. We want you to do your own due diligence. And this is my opportunity now to say, most important thing I have to say today is a reminder. You should never pay to participate in a clinical trial. If someone asks you to pay to participate in a clinical trial, don't do it. Don't go somewhere else. And the last thing I'd like to say is we recommend, whatever it is, if you're considering participation in a clinical trial, to please involve your routine eye health care provider for advice, bounce some ideas off of it. They have your best interest at heart. And also, if you have any questions about what is a trial a good idea for you, is it a legitimate sponsor? We are always happy to answer those questions here at the foundation. Always happy to receive emails about this to help your decision-making process. So with that, I will conclude my comments. And this last note gives me a great transition to say clinicians are here to assist. And our clinician today, our guest, is Dr. Abigail Fahim. She's not only a clinician, she's a clinician scientist. And he's an assistant professor in ophthalmology and visual sciences at the University of Michigan. I'm also happy to say that we have a chance to work with Dr. Fahim in other ways here at the foundation. He's a member of our scientific advisory board, and I work closely with her through the Foundation Fighting Blindness Clinical Consortium. So I'm going to turn it over to Dr. Fahim. And thank you, Dr. Fahim, Abby, for joining us on your Saturday.
Yeah, absolutely. Thank you so much for inviting me to speak. And Let's see. Can you see that?
We
Or
can,
can
Abby. Thank you.
Okay, good. That's actually not the first slide. Hold on just a second. There we go. That's better. Yeah, so I do have some slides, but mostly it's just a guide for talking about some of the issues, and I'll talk about the investigator perspective on clinical trials. I'll go to the next slide. I do have the opportunity in my job to participate in many levels. So I have a research lab where I'm doing the first step, which was talked about by the other two speakers, of basically target identification. So studying the disease at the basic science level in models, cell models, animal models, to understand why the retina is degenerating, due to a particular genetic mutation. And so that helps us identify pathways that we can target to change the course of disease. And then there are many steps after that: target validation, lead compound identification, optimization, then the preclinical trials of your drug or whatever it is in the animal models, and then the clinical trials and drug approval. And it's what I'm showing is an upside down pyramid, because there's attrition at every step. So, discoveries that we make in the lab at the basic science level, none of it's wasted. We use all of that information to learn more about the disease. But at every step, so a lot of those targets won't be validated, and then, some of the compounds won't be optimized, and then some of the preclinical models won't work out. And then of all the drugs that enter clinical trials, only 10% get FDA approved on average, across all fields. So every step of the way. So the higher risk is at the top, and so industry tends to get involved a little bit later on after some of it has been de-risked, and academic science labs are doing the first couple of steps. And so I get to participate at the top, and then I get to participate at the bottom in the clinical trials. And so that's I really enjoy that in my career. So how do we choose to participate in a trial? So at University of Michigan, we are a site for many inherited retinal disease trials, but not all of them. So first of all, the way it works is the sponsor approaches the sites. Those sponsors will approach sites and say, are you interested? And so we don't get approached by everybody, but we get approached by a lot of sponsors. And we say yes to a lot, but not to everything. And, we always, because IRDs generally don't have treatments, we really like to bring high quality trials to our center to be able to offer something to our patients. But here are some, so we usually invite the sponsor to present their data to us. We want to see what their data is. And the questions we have are, is it biologically plausible? Does it even make sense that this particular treatment or the pathway they're targeting would change the course of disease? What's the scientific rationale for that? And then in their preclinical data, which often involves, animal models, did it seem to be safe? And sometimes in animal models, yes, they do look at safety, but how well does that translate to human safety? So sometimes we also just think about expected safety, like if they're giving an intravitreal treatment. Well, we give a lot of intravitreal treatments. There's a lot of precedent for that. And historically, some intravitreal treatments have been very inflammatory, whereas others have not. So sometimes we can think about current treatments we know of and think about what is the expected safety of whatever the sponsor is proposing. And then preclinical efficacy. For us, that's kind of a big one. So one reason that, there are a lot of reasons that only 10% of drugs in clinical trials make it to FDA approval. But one reason is that the bar for having a clinical trial, the bar of showing efficacy, preclinical efficacy to get a clinical trial, is actually pretty low. The FDA to approve a trial cares a lot about safety. That's their number one. Is this likely to be safe? although, you never know 100%. That's why there's a phase 1 study to look at safety. But, in terms of efficacy, the sponsors really don't have to show that much to get a trial. And so sometimes a sponsor will show us their preclinical efficacy data, and it's really just not very convincing. And so then we tend to not do the trial. And then feasibility. So these are rare diseases, as you know, and so do we even have the patient population to recruit for this trial? And sometimes it's not just about the gene, like, do we have enough patients with this gene? Because there are other inclusion criteria, like they have to have a particular level of vision or they have to be a certain age. And so we look at our patient database, and not all patients want to participate in a trial. And so we find that we sort of estimate the number of patients that will probably be eligible, and then we sort of take a percentage of that and say, okay, well, this is realistically the number we could probably recruit. And if that's not consistent with what the sponsor's looking for, then we won't do the trial. And then recruitment, how do we recruit our patients? So for the trial. So this in some way is dictated by the IRB, which is the ethics board that controls sort of the ethics approval of our clinical trials. Often they have standards by which we are or are not allowed to recruit patients. So the most conservative approach is to send a letter. So you typically will send letters to patients in our database, meaning patients that have been seen at our clinic before, that based on their chart, they look like they might be eligible. I'll send them a letter and say, hey, we're doing this trial, contact us if you're interested, and we'll have contact information. Sometimes based on the IRB approval, we're also allowed to do a follow-up phone call. So, the approval for the trial will be something like send the letter and then say in the letter that you're going to call them in 2 weeks. And then so after we send the letter, we can follow up with a phone call and ask if patients are interested. Some sponsors can contact patients through my retina tracker, which is one advantage of being part of my retina tracker, which is the FFB research program for genetic testing. Sponsors can have my retina tracker send out letters. So the patient information isn't being shared with the sponsor, but the sponsors can indirectly contact the patients about trials for a particular genetic etiology. And then of course there's in-clinic recruitment. When patients come to see us, I always, share with them information about any clinical trial for which they might be eligible, both at our site and at other sites. I might tell my patients, hey, there's a trial going on at these other institutions. We're not a site, but you might be eligible for this. And I share the contact information on clinicaltrials.gov, which we just saw, and I'll give them that information. Sometimes the trials, especially in phase 1 and 2, the number that each site is approved to recruit can be quite small. So we've been a part of trials where we're approved to recruit 4 patients, for example. And depending on patient interest, sometimes patients get wind that we're going to be a site and they're calling us before we're even open to recruit. And so sometimes they fill very quickly before we even have a chance to send letters. But at other times, recruitment is actually quite challenging, and so it depends on the trial and the patient population. Okay, so these are some of the common questions that surround recruitment. I have them categorized by investigator questions and patient questions. So from my perspective, my questions when I'm thinking about recruiting a patient are, of course, does the patient meet the inclusion criteria for the study in terms of their genetic etiology, age, vision level, whatever else the sponsor has specified as a criteria? Is the patient likely to come to study visits? So if a patient has a high no-show rate, sometimes through no fault of their own, but the study visits, so often the visits have to be within a very small time window. And so if there's going to be a lot of no-shows or cancellations or rescheduling, that could be very challenging to have a patient in a study. Is this patient competent to consent to research? Some of our patients are syndromic and have some cognitive impairment. Of course, we want to make sure that they're competent to consent. And if they're not, is there IRB approval for their legal guardian or power of attorney to consent for them? So that's always an important question. And is the patient able to reliably complete study tests and assessments? You know, this comes into play a lot for some of the trials that are recruiting children. And, you know, some, depending on the age of the child and how well they're able to do study tests, sometimes, you know, that's definitely an important consideration. This also comes into play sometimes with cognitive disability. You know, if the study requires visual field tests and the patient just isn't able to cooperate with those or understand the tests, some of those tests can be quite challenging. So we need to make sure they're able to actually do the tests required. And then the patient questions. Number one question I get is, does the treatment work? And unfortunately, it's not a question I can answer because it is a clinical trial, which means it is an experiment. So we do not know. And also, I think a lot of times patients, you know, maybe we've already recruited several patients and the trial's been going on for a while and the patient wants to know, well, has it been working so far? And that also is not something that we can share or even necessarily that we know. I mean, a lot of these studies are multi-site studies. We might have 3 patients, but overall maybe 20 patients have been enrolled. And the sponsor doesn't share like the information about patient, you know, about the clinical course for the patients during the trial. That data is owned by the sponsor and it's not like shared with all the investigators. So does the treatment work? It's not a question we can answer, but we can answer if we think it's, I mean, we wouldn't have the trial at our site if we didn't think it was biologically plausible and if we didn't think the potential benefit outweighed the potential risk. But that's pretty much as good as we can do. Is it safe? Again, it depends on the phase of the trial, whether we can answer that or not. Phase 1 studies are to determine safety. So if it's a phase 1 study, we don't know if it's safe. If it's phase 2, phase 3, we can say, well, based on the phase 1 study, we think that this is safe. But you know, now that we're doing larger numbers of patients, it could be there are more rare side effects that we are going to find out. But you know, like for example, if it's a gene therapy, we've done a lot of gene therapy trials, and generally that is safe, although there are risks to any surgery. So we can't give a complete answer, but we can give some answer to that question. And then what is the time commitment? And if the patient doesn't ask that question, they should. And it's something that I said, well, think about the time commitment, because depending on the study, there are lots of visits. The visits take a long time. There are a lot of assessments, and so it is a time commitment. And this, I think we are kind of already went over the next 2 slides, but these are websites I share with my patients, the ones that we've talked about. The Foundation Fighting Blindness website, where under the research tab, you can go to the clinical trial pipeline and see the trials that the FFB lists. The FFB site of clinical trials is very user-friendly and patient-friendly. The clinicaltrials.gov is the most comprehensive database, but can be a little bit harder to navigate. But Todd went over that very, very nicely. And current clinical trials. So these are trials that we're currently involved in. We were a part of we are a part of the RPGR clinical gene therapy clinical trial, which is completed and is in discussions with the FDA. So we're all kind of waiting to hear about that. But these are other trials that we're currently involved in. And my toolbar is getting in the way of, there we go. I think it was just in my way, not your way. So we're part of the NAC-ATTACK trial. This is N-acetylcysteine for retinitis pigmentosa, a multi-center study. So this is actually an over-the-counter supplement that is approved for protecting the liver in cases of Tylenol overdose to prevent liver damage. And it in a small study, it showed some improvement in macular cone function in retinitis pigmentosa. So now there's a large multi-center study, a 4-year study looking at N-acetylcysteine, pretty large doses of N-acetylcysteine to slow progression of RP. It is placebo controlled, so 2/3 of patients get the N-acetylcysteine and 1/3 get placebo, which is the best trial design to really know if the treatment works. And it's unique in that it's a 4-year trial, and that's because it's funded by the NIH. Most trials are funded by pharmaceutical sponsors, and it's really tough to pay for more than a 2-year trial. And that's really one of the problems that we've run into with IRD trials, because it does take longer than 2 years to show that you're slowing down progression of a slowly progressive disease. But this one's NIH funded. It's a 4-year trial. We're also part of the Ascidian trial for Stargardt disease. You can see 3 trials for Stargardt disease we're involved in: Splice Bio trial and Metformin trial, and then a trial for PRPF31 related RP. And I actually have some slides to kind of talk about some of these therapeutic approaches, which are a little bit new and different, some of them. So this is just a slide looking at some broad categories of different types of clinical trials for IRDs. On the upper right, I show gene augmentation. This is sort of the traditional, I say gene therapy is still relatively new, but this is quote unquote traditional gene therapy, where you're basically doing gene replacement, putting in a normal copy of the gene. And this has been used in RP and LCA and achromatopsia clinical trials. There was a gene editing CRISPR trial for LCA, Leber congenital amaurosis. And then RNA, ribonucleic acid, is a very versatile type of molecule and is being used as a therapeutic for clinical trials in a variety of ways. I have listed here Usher syndrome, LCA, RP, and Stargardt. Many of you may know this, but our DNA, of course, has our genes, and then that's translated into these temporary RNA messages. And then the RNA gets made into a protein. And eventually the RNA gets degraded. And so the RNA and the protein is really what carries out the job of the protein is the business end of it that really carries out whatever functions the gene was meant to do in the cell. And so that temporary intermediate, the RNA, is a very versatile molecule, and RNA tends to bind to other RNA. So RNA is made up of nucleic acids that have a particular spelling sequence. And if it encounters another RNA that has the opposite sequence, it binds. And so if you have a particular RNA message that you want to impact, you just design an RNA that has the opposite message. And this has been used in a variety of ways. And then there are drug trials for Stargardt and retinitis pigmentosa. And then RPC stands for retinal progenitor cells. There have been trials for those. Those are fetal cells that are kind of halfway between stem cells and photoreceptors. Not really the same thing as stem cell therapy, which we'll talk about in a second. And then optogenetics, where light sensitive, it's basically a type of gene therapy that makes a light sensitive protein to make the remaining retina cells more light sensitive. So, you have your rods and your cones, which sense light and send vision to the brain. And then you have other types of neurons in your retina that don't normally sense light. And this optogenetics type of gene therapy makes some of those cells light sensitive to it gives a type of vision, like flashes of light in the shape of whatever you're looking at. So more for end stage disease. And so this is just a slide as an example about voretigene neparvovec, the one approved gene therapy that we have for inherited retinal disease, just showing how it's delivered. It is delivered by, and this is how gene therapy in general is delivered, by an eye surgery where the gene therapy molecules are injected underneath the retina. And then the DNA that's packaged in here gets inserted into the cells that need it, the retina cells that need it. I just have a few more slides. So this is just, I'll just describe what's in this slide. So the first CRISPR clinical trial for IRD was for a gene CEP290 that causes Leber congenital amaurosis. It was a phase 1/2 study, and the results were somewhat variable. So 6 out of 14 patients improved in the first test that's shown at the top, which is a kind of test FST, which tests the sensitivity of the retina. And then 4 out of 14 patients improved in visual acuity. 4 out of 14 improved in their mobility test in navigating. And then 6 out of 14 improved in a quality of life questionnaire. So kind of some improved, some didn't. There was some somewhat variable success there. Patients that had the best baseline preserved structure of their retina showed the largest improvements. And in general, that's what we think is if we can intervene earlier in disease, that there's a better outcome. So talking about RNA therapies, so one of the trials that has been exciting is for USH2A, Usher syndrome and retinitis pigmentosa. And this is an RNA therapy that initially was run by ProQR, and then the trial was unfortunately stopped, but the product has been licensed by Thea, and there's a site in Texas recruiting. And this is an RNA molecule that binds to the RNA message for USH2A and skips over mutations in a particular region of the gene called exon 13. And so patients that have mutations in that region of the gene, this RNA therapy will help the protein skip over that entirely. And there's another type of RNA. So this is one of the trials that is upcoming that we're involved in for PRPF31 related retinitis pigmentosa. And this is basically, there's a molecule C0.3 that reduces expression of PRPF31, and this RNA molecule decreases C0.3 to indirectly increase PRPF31. So just another kind of creative way of using RNA to treat a disease. And this is one of the trials we're involved in for Stargardt disease. One of the problems with Stargardt is the gene is so big, it's too big to fit into our traditional gene therapy vectors, as many genes are, like USH2A. And so sponsors and researchers are coming up with creative strategies of how to get around that. So this is an RNA molecule that encodes the first half of the ABCA4 gene, and it binds to the patient's RNA message for ABCA4 and replaces the first half of the patient's message with this new correct message. So it's an editing RNA. And this type of technology is exciting because if it works, it can potentially be used for other genes that are too large as well. And there's one more for ABCA4. We're also a site for this one, which is up and coming, which is another approach for genes that are too large. So this is also ABCA4, and basically there are 2 gene therapy vectors, one for the first half of the protein and one for the second half of the protein. And so they make these 2 halves of ABCA4. And how do the 2 halves get together? So this technology is taking advantage of something called inteins, which are derived from cyanobacteria. So cyanobacteria make these inteins, which are these tiny protein peptides that are binding partners. And when an intine finds its binding partner, it binds and they splice each other out. So in these gene therapy vectors, each half of ABCA4 has a tiny little intine on it, and they're binding partners. And so the 2 halves find each other and the intines splice out, and you get a full-length protein. Again, if this is something that ended up working, that could potentially apply to other genes that are too large for traditional gene therapy. So a lot of creative science going on and creative clinical trials. And then what's very exciting is stem cell therapy, and we heard about David Gam's work. So we can make stem cells from any of us, from our skin, from our blood. We can make pluripotent stem cells, and we can turn those stem cells into rods or cones. And a lot of the work is focused on cones, even though, for example, in RP, rods are lost first, but cones are really thought to be the most important for the highest resolution vision. So there's been a lot of emphasis on making populations of cones. And then ideally, these cones would be transplanted into patients with retinal degeneration to replace the photoreceptors that have been lost. This has not been done before. There's been so much about stem cell treatment that sometimes there's a misconception out there that we're already doing this, that we're already transplanting stem cell derived rods and cones. We are not. There has been proof of concept in mice that this can improve the visual behavior of mice with retinal degeneration. And we're still waiting for the first clinical trial with this type of technology. And hopefully it's coming not too long from now. So this was an FDA consumer alert that because there have been a lot of kind of rogue stem cell clinics, and the FDA, it says the FDA has the authority to regulate regenerative medicine products, including stem cell products and exosome products, which is exosomes are little vesicles that are released from cells. It says there's a lot of misleading information on the internet about these products, including statements about the conditions they can be used to treat. FDA is concerned that many patients seeking cures and remedies may be misled by information about products that are illegally marketed, have not been shown to be safe or effective, and in some cases may have significant safety issues that put patients at risk. FDA wants to help consumers be informed about how these products are regulated and what to look for when considering treatment with one of these products. And then they go on to say that these products are approved for use in patients with disorders that affect the production of blood, but they are not approved for other uses. So currently, stem cell products are only approved to treat like blood cancers, basically. But hopefully, in the not too distant future, we'll have these types of treatments for eye disease. And this is a paper from 2017. This is one example of an unregulated clinical trial for retinal disease that turned out badly. And this was a trial for intravitreal injection of autologous stem cells for age-related macular degeneration. For a while, a lot of clinics were taking cells from patients like fat and stem cells from patients' adipose tissue and injecting them into their eye. There were some rogue stem cell clinics. These cells actually cause a lot of inflammation. And so these patients on the right, I'm showing a picture of a patient with basically a total retinal detachment. And on the left is a patient with diffuse retinal hemorrhages and retinal vein occlusions that caused extreme vision loss. So there are dangers. And so you want to be very careful. I'm showing there are these predatory trials that aren't really real or approved clinical trials. And this is one of the things that Todd Durham was saying, if you're being asked to pay for a clinical trial, that should not be the case. That's a red flag, because sponsored clinical trials, they pay for your treatment. They usually even pay for travel expenses, not always, but often they do. If the trial is not registered on clinicaltrials.gov, and it's just a question mark, you need to look into that more. And if all of the preclinical data is on their website without peer-reviewed publications, that I don't know. I have that on there, but sometimes the data might be proprietary. So I guess that's not necessarily a definite red flag, but you need to talk to your eye care provider before participating in any of these trials. That would be the best course of action. So with that, I just thank everyone for listening. I might have gone a little bit long, but that's all I have.
That's great, Abby. I loved how you covered so much material in your presentation, new technologies, clinical trials, how you think about clinical trial opportunities as an investigator. And importantly, the point we want to emphasize is there are examples of trials you shouldn't be involved with. Things could not end very well for you. So I want to, this is Todd Durham again. I want to change directions one more time and to expand our presentation today. We want to include the perspectives of individuals and parents whose children have participated in clinical trials to bring in their perspective. So I would like to introduce them all at once, and then they're each going to take a turn to tell a little bit about themselves and their experiences. So up next, we have Lindsay Rambo, Paula Dubecki, and Judy Peckham. Welcome.
Hello, everyone. My name is Lindsay Rambo, and I just want to start by thanking our hosts for holding this webinar and inviting me to participate as to share my experience with a clinical trial participant. I am currently participating in the LCA 5 clinical trial at the University of Pennsylvania. And as a background, I was diagnosed with disease of the retina at 8 weeks old and was prescribed glasses at 10 weeks old. And I went through the majority of my childhood just kind of using the bare minimum as far as accommodations go, basically just wearing my glasses and using a handheld magnifier. I struggled in my teenage years as I was not able to drive a car because of my legal blindness. And I also received the news when I was 16 years old that I would probably be totally blind by the time I was in my early 20s. And if I ever had children, they would be blind as well. So following that news, I was not motivated to go back to an ophthalmologist for some period of time. Within that time, I met my husband, Zach, and he was a light in my life and kind of helped me grow to accept my vision condition, because I very much hid my vision condition from everyone else and tried to appear as normal as possible throughout most of my life. So he definitely helped me embrace my vision condition and see it as a positive rather than a negative. And one of the ways he did that was by helping me connect with the Foundation Fighting Blindness, and specifically helping me get to the University of Pennsylvania in 2013, where I met with Dr. Jacobson for the first time and underwent 12 hours of testing at the Shea Eye Institute to learn that I had a rare form of retinitis pigmentosa that was later diagnosed as Leber's congenital amaurosis 5 when I was 29. Some time passed from my first visit with Dr. Jacobson, where he was the first doctor to ever tell me hopeful news at the end of a visit, and where he told me that I would likely be able to experience some sort of treatment within the next 5 to 10 years. And I was referred to the National Eye Institute at NIH for several years, because I wanted to go somewhere a little bit closer than Philadelphia to where I was living at the time. And that is where I underwent the genetic testing, and they learned the deficient gene was the LCA5. And from that point on, my husband continued to monitor and do his own research to see if there were any clinical trials being conducted for my specific gene. And in January of 2022, he found Opus Genetics clinical trial that was going to be conducted at the University of Pennsylvania, kind of where my whole story began as far as getting a diagnosis. And we contacted the research coordinator there and sent over all of my records and everything from NIH. And then in June of 2023, I was enrolled as the first participant in the LCA 5 clinical trial. And then in September 2023, which was last year, I had my treatment. in my left eye, and I just recently had my 1-year follow-up from my surgery, and my left eye, which was once my worst eye, is now definitively my best eye, and my central vision has increased by 10 times. I have been able to navigate in dimmer light compared to my baseline testing prior to the surgery, through the virtual reality orientation mobility course, and many other surprising little life kind of things that I would notice in my everyday life. And just one more thing I wanted to mention is that I have had vision throughout my life, which is very rare for this type of disease, because many individuals affected by LCA do tend to lose most of their vision at birth, and I am still able to visually read. I know Braille. I taught Braille for 3 years. However, I do not use Braille personally. I read with screen magnification. So I have been very blessed. I turn 36 next week, and I'm thankful to still have the vision that I do have, and I'm thankful to report that the clinical trial has been a success in my own life. And I thank you all for your time and listening.
Paula, the floor is yours.
Thank you and good afternoon. My name is Paula Dubecki, and I'm here today as both a parent and a caregiver. I'm deeply honored to share a little about my son's journey and the role I play in supporting my son, Marty, who is currently enrolled in a clinical trial. So to give you some context, our family's journey began when my son was diagnosed with X-linked retinitis pigmentosa at a young age. As you might imagine, it wasn't easy hearing the diagnosis. As parents, we felt fear for the unknown. How much vision will he lose? How quickly will he lose his vision? How will his vision loss affect his future? We have both concern for his future. We have both concern for his future and determination to do whatever it takes to provide him with the best possible care and opportunities. As the parent and clinical trial caregiver, my primary role is to ensure that he has the support, encouragement, and resources he needs to navigate the world despite his challenges. This means I accompany him at medical appointments, understanding his condition, and encouraging him to advocate for himself as well. After being turned down for one clinical trial, we were disappointed, but then he was accepted into the MGT RPGR clinical trial. Like many families in similar situations, we were searching for hope, a chance for something that could make a meaningful difference. Enrolling him in this trial represented not just hope, but a proactive step toward a brighter future. It is an opportunity to contribute to research while also potentially improving my son's quality of life. Being part of this trial has brought unique responsibilities. Beyond caregiving, I find myself arranging transportation and lodging for doctor's appointments. It is both challenging and rewarding knowing that our participation may not only benefit my son, but could also pave the way for advancements that help other families in the future. Through it all, I've learned that caregiving is as much about resilience as it is about adaptability. There are good days when progress feels tangible, and there are tough days when uncertainty looms. But every step of this journey has reinforced the power of community, science, and the human spirit. I'm grateful for the incredible team working on the trial my son is enrolled in, as well as other researchers dedicating their lives to finding solutions, and for the other families who inspire us with their stories of courage. Together, we're part of something much bigger than ourselves, a collective effort to push boundaries and create possibilities. Thank you for giving me the opportunity to share our story. It's a privilege to present not only my son, but also the many caregivers who are walking similar paths. I look forward to the hope and progress clinical trials will bring for all of us. Thank you.
Thanks so much, Paula. Jody, the floor is yours.
Thank you. Good afternoon, everyone. I just want to say thank you for everyone for attending and for giving the opportunity to speak this afternoon on behalf of my son, Jacob. Like Paula, my son was enrolled in a clinical trial, and it was definitely a unique situation on how we got there. And I want to thank everyone that's been in my village. You know, it takes it takes a lot, and to have the support and the guidance. Sometimes it's not easy. It's not that easy street. When Jacob was first born in 2012, we were told he has nystagmus and that his eyes just wobble. So after going every six months to the pediatric ophthalmologist, we then were switched to a couple doctors here and there. And what I'm learning through this all is that you're not always guided directly to where you need to be. And sometimes you have to have these hurdles and these bumps and these speed bumps to get to where you need to be. So it wasn't until I was at Jacob's IEP. And for those of you who don't know what that is, it's basically a meeting at the school with the support staff. So Jacob had services. So he had a TCVI, teacher for the visually impaired. He had a O&M, he had PT and OT. He had all these services for his nystagmus and his different eyes. And with that, his orientation and mobility, who also had a background in TCVI, said, well, why don't you go to Michigan Medicine? And I said, I can't go to Michigan Medicine because I went to Michigan State. And for those of you who aren't living in the mitten, you know, you're either team U of M or you're team Michigan State. So it wasn't until that conversation, sitting in that I think it was his second grade or his third grade classroom, where I said, you know what, let's just do it. So blessed by the people at the University of Michigan that we were able to connect the dots and be on the path of a similar situation like Lindsay, all the way over to Philadelphia. So unfortunately, we weren't able to stay at Michigan Medicine for the clinical trial, but they were the stepping stone that got us to where we needed to be, which was in the University of Penn Medicine, and we worked alongside with CHOP, the Children's Hospital of Philadelphia. So there were a lot of moving parts, but I want to say, you need to have the people and you need to advocate and continue to say, I'm not going to give up. This isn't just nystagmus. This is more. And was it scary signing all those papers? Absolutely. But at the end of the day, I said, well, what have we got to lose? Jacob's either going to continue to not be medicated and not go through this gene editing, or we can try this and maybe it's going to work. So and I'm a believer of faith, and so I said, you know what, we're gonna pray and give it to God at this point, and let's go from there. So, and everything happens for a reason. And I'm a true believer in that. Even though it was the time that I was completing chemotherapy treatments for breast cancer, I was in the midst of getting a new job. Now I'm signing papers for my husband to take my 9-year-old to Philadelphia. Like, there was just a lot going on. But I am here, 2024, 3 years after this clinical trial began, to announce to all the participants and all the attendees on this webinar that it's a success. And I am so grateful. I'm so grateful for the village and so grateful for my people that are cheerleading and saying, you know, you got this. That when Jacob says, am I going to be in the science books? You know what, Jacob, you might be in the science books one day. So again, I know it's a lot of unknowns, very scary, but please make sure that you don't give up on advocating for either yourself or your loved one, and being that cheerleader and having that medical team support you. And if you have a gut feeling that something's wrong, then find someone else. There are people out there like Dr. Fahim, Dr. Durham, other people that are in society that are for helping and creating a wonderful outcomes. So kudos to those in the medical field and all the science and research that has allowed us to be where we are today. So on that note, I'm going to go on mute. If anyone has any questions, concerns, any of that nature, please feel free to reach out to me. And I hope everyone has a great rest of the weekend. Thank you.
Wonderful. Thank you so much, Lindsay, Jody, and Paula, for sharing a little information about yourselves and your stories. So I know we're running short on time, but we are going to go ahead and move forward with the Q&A portion of the program. So to kick us off, we're going to start by answering some of the pre-submitted questions, but feel free to drop any additional questions in the Q&A function. We're going to get to as many questions as we can in the next 10 or so minutes. But if we do not get to your question, do not worry. We will have a staff member who will reach out directly to you. So I'm going to go ahead and pin our panelists here. Abby, I'm going to go ahead and let you know that you are going to be up first to bat. So if that gives you some time to get yourself ready. So one of the first, perfect. So one of the first questions that we received is, how unusual is it to not have a noticeable peripheral vision loss until late 40s for someone who has USH2A?
It's not that uncommon. I mean, if we formally tested your visual field, you would have a defect before age 40. But these diseases happen slowly, and some patients notice the visual field loss more than others. I have had patients who are legally blind by visual field who are still driving because they just haven't noticed what they've lost. And it's actually very surprising to them that it comes as such a shock when I have to tell them, unfortunately, that they shouldn't be driving. So it's not uncommon at all. Your brain adapts so well and it fills in. When we see these RP simulations, online, it's always like a tunnel with like black around it. But that's not what the patients see. I mean, that's what they tell me. It's not that they see black. If I hold my hands back here, I can't see them, but it's not that I'm seeing black. Your brain pretends that what you're seeing is your whole visual field, is the whole visual field, is what I'm trying to say.
Perfect. Thank you, Abby. So this one I'm going to throw over to either Todd or Abby, whichever you all want to take this one. I am an 87-year-old with dry macular degeneration. Would I qualify for clinical trials? And I will say we do have a few questions around qualifying for clinical trials, so I think feel free to broaden this one out a little if you feel fit.
Yeah, I'll take a first pass at this and then ask Abby to fill in too, because she's going to have experience with a number of study protocols. But my sense is, even from the research that we do at the foundation, there's no age requirement. Your chronological age, probably what's more relevant is what is the status of your retina, either the structure or the function, for that particular study. So just this person's age wouldn't necessarily, in most cases that I know of, would not be disqualifying. But there might be something else, other eligibility criteria that you would need to meet. Abby, is there anything else you'd add?
Yeah, I would agree with that. I mean, for many of the IRD trials, there is an age cap. You know, there'll be an age range. Some of them don't put an upper age limit, and some do. But for age-related macular degeneration, since it is an age-related disease, most of the trials don't have an upper limit on the age. But yeah, it has more to do with sort of where you are in terms of your severity and the geographic atrophy and things like that.
Thank you. And Amy, I'm going to throw this one over to you. So how far along are we with gene therapy?
So there is an approved gene therapy for an ocular disease on the market. Dr. Durham mentioned the Luxturna gene therapy for RPE due to mutations in RPE65. There are a number of clinical trials, and we have them listed on our website as well, that are looking at potential gene therapies for a variety of retinal diseases. Additionally, there's researchers that are optimizing the tools to conduct gene therapies. So we're really along the entire developmental process for gene therapy, from discovery of new tools all the way through market approval.
Perfect. Thank you. And Abby, I'm going to throw this one back to you because there was a question around, on the clinical trial for IRD slide, which LCA is being treated in those bubbles? And we can also share that in the follow-up as well, if that is easier, so that you don't have to go back to your slides.
Yeah. Well, so definitely on the gene editing slide, I was referring to that CEP290 trial. Gene augmentation, there have been a number of LCA trials. We just heard about LCA5, and of course RPE65. Off the top of my head, I'd have to look back and see what other genes have been. Not all of the ones in these bubbles are like actively recruiting currently. I'm sort of listing past and present applications of these. LCA for RNA, LCA, I think CEP290 at one point had an RNA therapy. Yeah. So sorry, I don't know how helpful that is. For currently recruiting trials for LCA, I would definitely use the clinicaltrials.gov site with, if you put in Leber congenital amaurosis or your gene of interest, if you have a particular gene in mind, and then click on the actively recruiting and interventional. Those are like two of the filters that are most helpful.
Perfect. Thank you. And we will make sure that after the webinar, everyone has the link to clinicaltrials.gov as well as any of the supporting information. That way you don't have to go Google it. So I'm going to switch gears a little bit for the last couple of minutes here, and I'd love to hear from Lindsay, Jody, and Paula. So we have had a couple questions. First off, a lot of people who are appreciative of you all sharing your personal stories and your family's stories related to this. So one question we had in here, and I apologize, my chat keeps moving. For those who went through a trial, what are some of the things you can do or see now that you couldn't before? So Lindsay, I know you personally went through. Jody and Paula both had children. So whichever wants to start first.
I don't mind sharing. I think the biggest thing that I started noticing once like the air bubble had subsided and everything after the surgery, and my pupils had not been dilated anymore, was just like clarity. I could tell that colors seemed so much more bright to me. I think a big one was being able to take the text size down on my phone, actually, because I used to have it like super big when I was reading, and I like went down 1 or 2, and it still remains to that point. And also just like the screen magnification. I remember the first time I was sitting and looking out my window, and I still get a little emotional about it, but I could pick out my own children playing outside in a group of kids, and I'd never been able to do that before. So that was a really big one for me. Um, and just little things in my everyday life, like I didn't have to use a white cane anymore. I was able to kind of navigate in unfamiliar terrain. If the lighting was welcoming to me, I still struggle with transitioning and different lighting and everything. Um, and I'm not like colliding so much with things that I used to run into around the house, or just little things if I left a cupboard door open, or you know, missing the doorway, kind of turning in and hitting the side of the doorway. Like I'm doing that less frequently now. And that all kind of aligns with what they're seeing and all of the testing and everything and at the University of Pennsylvania.
Perfect. Thank you so much, Lindsay. And Jody, we had a question directed towards you. So
Sure.
one of the attendees asked if you could share which condition your son had. Was it RP? And if so, what gene?
Yes. So CEP290 was the gene, LCA. So yes. So he, and like Lindsay, it was, LC10 was the trial name. So, I don't know if that's the pediatric version of, the young adult version. But yeah, and how ironic that it was at the same, the same hospital. And then likewise, I wanted to echo what Lindsay was saying. My son, the same thing. At that time that he came back, he from baseline and all of that, same thing with his iPad. He took the magnification down and his text down, and I'll never forget. And gosh, I want to get choked up about it because it's so exciting. Is he showed me his iPad and the text was smaller, and he's like, "Mom, I could see this," and it was just such a moment that we celebrated. I took a picture of him holding the iPad with the text smaller, sent it to his group, his support people, and just said, this is what's happening. So thank you for the opportunity of sharing that information and that little tidbit.
Of course. Thank you so much. And it really is a testament to the hard work that all of our researchers and scientists are doing in the lab, that, being able to know the real world reaction that this is having on people's lives. So thank you all so much for sharing that. So I know that we are a minute after. It's 2:00 here on the East Coast, but we will be following up with everyone who we are not able to answer their questions today live. We have recorded this session, so we will be sure to send that out as well in the coming days. If you enjoyed today's webinar and you'd like to get involved with other activities at the foundation, such as our vision walks, our vision connections, or any other local activities in your area, please feel free to reach out to us at info@fightingblindness.org. Once again, I want to thank our amazing presenters today for sharing all of this great content, and we hope you have a great rest of your Saturday. Have a good one, everyone.